quick video going over transcription, translation, and what it means for the expression of DNA to code for genes and traits. Now, a quick overview of some things that we talked about last week and on Monday or Tuesday is DNA structure. And the reason why I think it's important is to understand DNA structure, to understand DNA structure, you'll be able to understand why and how production of proteins is specific to each of the 30,000 genes in the human body. Now, when we look at the structure of DNA, we have nucleotides that are both purines and pyrimidines. We know that one purine and one pyrimidine, a purine would be adenine and guanine, pyrimidines would be cytosine and thymine, form triple or double bonds. So we could think about DNA as either big or small, 3 or 2, when it comes to the coding strand of it. We know that when it comes to DNA base pairs, that we have C's and G's forming together to make one rung of the ladder, and they need 3 bonds. And A's and T's need 2 bonds. The low and the high maintenance relationship couples, as we said, for DNA. So when we read DNA, we can read it as a code as either big or small, 3 or 2, when it comes to the single side strand that's codes. So again, DNA can be big, or small, 2 or 3, 3 or 2, when it comes to how we look at it. So let's get into translation and transcription. So transcription is going to be the process of copying our DNA from one side of our DNA to make mRNA. And mRNA is just a single temporary copy of our DNA that is going to be leaving the nucleus and going out to find ribosomes in the endoplasmic reticulum, and then that's going to be used to make our proteins. So transcription is where we make a single temporary copy of a gene, and that allows us to express proteins in a variety of ways. It helps us regulate protein production based on time, so genes can be turned on or off. It helps us regulate the amount of proteins that we make. RNA can be copied 1 time or 1,000 times. And it helps us regulate what proteins are made in which cells. Remember, you don't want hydrochloric acid made in your eye cells. You want to make it in your stomach cells. Transcription allows us to do it where we copy the gene into a single copy of RNA. Now, translation is where we take our RNA and we are making a protein out of it. And this is where the shape and sequence of DNA is super important. mRNA is going to be read by an enzyme called tRNA. We can see them right here. Notice blue, blue, red, yellow, red, red, and red, blue, green. Each of these is a complementary attachment that can recognize only A, T, G, or C. And that's where the shape, big or small, 3 or 2, is really important. Each type of tRNA, these goofy little green guys that we see right here, only binds to one specific amino acid. So each type of code, 1, 2, 3, codes for a specific type of amino acid. And we get that from the specificity of what are the 3 nucleotides in this codon? Are they big or small? Do they code for 3 or 2 nucleotides? Every 3 nucleotides is what we call a codon. So transcription occurs in the nucleus. RNA leaves and goes out to the ribosomes in the endoplasmic reticulum, and then translation is where we build our protein. So let's talk about this. The tRNA molecules that we see right here are the enzymes that bind to our 3 codons, and they attach to our RNA temporarily, and that's how we build a protein. Each codon codes for 1 amino acid, so a sequence of DNA that's 9 nucleotides long would code for 3 amino acids. Now, in terms of the order of codons, the first nucleotide is the most important. It can be G, A, T, or C. So this is going to be determinant of what type of amino acid you fit into. The second amino acid, which we go from G to C, T, or A, or G, is also very important. These 2 are the most determinant of what type of amino acid codes. Generally, we find that the third nucleotide or the wobble nucleotide does not matter for whether or not you're alanine or valine. So if I'm GTT, I'm going to be valine. If I'm GTC, I'm also going to be valine. This is because when we look at the tRNA, well, it's not shown. It's almost like a wave. The first, then the second, then the third nucleotide in a tRNA binds. So that wobble codon, that third nucleotide, is the least important in determining our amino acid. Now this is important because when you start to make changes to the code, we start to see issues. So for example, if we look at the DNA for a normal gene to make hemoglobin, we tend to have a codon that is CTC, and that creates a very vibrant red, robust red blood cell because of this glutamine acid. However, if we go from CTC to a CAC, we're swapping that second nucleotide in the codon. We switch the code and now we're producing a valine. And a valine is a hydrophobic amino acid which causes the protein shape to change and fold inwards, causing us to have the sickle cell shape, which is not a red, robust, large hemoglobin, but rather a small, shriveled one up. So we tend to say that the first and second nucleotides are super important. The third tends to be less important. Tomorrow we're going to be practicing making proteins with our DNA codes and also thinking about different types of mutations. For now, I think it's really important that you understand what a codon is and how the sequence of DNA is really important for what amino acid codes in our nucleotides. I hope this has been helpful. Let me know if you have any questions, and I will see you tomorrow in class.